Okay, I think the hush over the crowd and the music stopped. That means it's time to get started. So welcome everyone. It's great to see a full house here on this cool rainy evening. My name's Aaron Marshall. I'm a professor at the Department of Immunology at the University of Manitoba, and it's really a pleasure to have this opportunity to moderate this discussion on the topic of immunology research from a graduate student perspective. Before I tell you anything about myself and immunology research, just a little about what this event is. And it is intended to be a discussion. We're going to spend most of the time having an opportunity to interact between you guys and our students and myself up here. I'm going to try not to lecture, but that's what I do for a living, so you may have to shut me up. So Café Scientifique, I am told, started in the late 20th century, something like that, 100 years ago, as with this concept of engaging scientists, not being these evil guys out there in white coats, but as members of the society, engaging with people about the research that we do. So this is a CIHR, University of Manitoba CIHR Café Scientifique. So it's a mouthful, but now I'm going to expand CIHR to tell you what that is. That stands for the Canadian Institutes of Health Research, which is an important organization funded by your tax dollars that funds research into health issues that affect Canadians. And in fact, all the research projects that are going to be touched on tonight were funded by CIHR. Now, I have a couple of housekeeping things, and I'm going to refer to my list because I won't remember them if I don't do that. First of all, I'll just give you the names of our panelists, and I'm going to introduce them individually as their turn to speak. We have Wiebe, Samantha Pauls, Natascha Fitch, Sam Mosova, and Rebecca Schneider. They're all, again, graduate students in the Department of Immunology. Be kind to them. This is actually kind of a unique twist on this cafe concept that we're doing tonight by having essentially scientists in training talking to the public, which is something that they usually aren't given the opportunity to do. But we felt that they could be some of the best spokespersons as individuals who really immersed in the trenches, which in this case is a biomedical research lab, trying to make discoveries to further health research. These would really be good folks to interact in this kind of session. I'm told this night's session is going to be live on Twitter, which can be accessed using the hashtag @CaféScientifique, all one word. It's also being videotaped and will be available on the web afterwards at manitoba.ca/research/CaféScientifique. Could be on the radio. So we're going to have each of these folks speak for about 5 minutes, and after that we'll have our question and answer session. And I'll also point out, if you haven't noticed already, there are refreshments and some materials about immunology research in Canada over at the table here, and I think there are surveys for providing feedback about the session that we'd like to hear what you liked, what we could improve about the session at the end. And last on the housekeeping list is just want to emphasize that we really ask the audience to refrain from asking personal questions about their personal health situation and treatments. Again, we're here to talk in a general about issues that are of common interest to all of us here, and we're not aiming for those sort of personal health questions. Okay, so a little bit about me, and again, I do scientist that does research in immunology, and a good part of what I do is actually mentoring folks like or up here, which you may or may not know. How does a scientist become a scientist? Well, normally they will be in a graduate program, mentored by a scientist who's already an active researcher and essentially learning how to do research, and by working in a group like that. So I think what you need to appreciate, and I want to give these guys as much credit as I can. So I'll use an analogy. My kids liked dinosaurs when they were little, a little bit more grown up. now, but one of my sons wanted to be a paleontologist. And, you know, it's a grand thing to put together dinosaur skeletons. There's one back there, a real one. But what does it take to put together a puzzle like that? If for identifying new species of dinosaur, it involves a lot of grunt work. You have the site, you have the concept of what this new dinosaur species might look like, and then painstakingly piece together the puzzle over the course of years. And being a graduate student is doing a lot of that work of painstakingly piecing together the puzzle bit by bit by bit. And this is how we learn to be scientists. But these guys also have a great grasp of the whole, what the big picture looks like, in addition to the piece that they're working on. And I think it's going to be a treat to have the chance to hear them speak. My own interest in immunology, in 2 minutes, is really driven by two things. One, that this is just such a powerful thing. If you consider all the bugs that are out there, that we have this system to keep us healthy, and it's such an amazingly complex task because there are many threats, and most of the time it works. And the second thing that really fascinates me, maybe even more in recent years, is when you think about the chronic diseases that are really prevalent in our society today. The immune system has a lot to do with those as well, and in many cases, the immune system is not having a beneficial effect in those diseases. And so these are cases like rheumatoid arthritis, where the immune system is destroying your joints, lupus, where the immune system is generating a response against your whole body, essentially. Type 1 diabetes, which destroys the islets that produce insulin, et cetera, et cetera. So there's a lot of chronic diseases where sort of negative side of the immune system is revealed, and that, for us as researchers, is important because we spend a lot of time thinking about the molecular controls that turn immune cells on and off. And clearly it's a powerful system for good, but we need ways to be able to understand why the immune system goes wrong. And actually a lot of what you're going to hear about today in terms of the health applications of the research are in situations like that of chronic disease, where the immune system is having a destructive effect, and we need to understand why that is so that we can do something about it. Okay, not too bad for a guy that's lots for a living. So I'm going to start with our first speaker. Make sure I get all his background details right. Chris Wiebe, on my far right here, obtained his M.D. degree from the University of Manitoba, and he's a practicing clinician, the only one on our panel today. He's currently pursuing a master's degree researching factors affecting rejection of kidney transplants. Chris?
Great, thank you. As Dr. Marshall said, I work at the Health Science Center in kidney medicine, and my specific area of interest is kidney transplant, which, as I think you'll see by the end, is directly related to immunology. And as many of you may know, kidney disease in Canada is actually a really hot topic, a really important one, because if you look at the last 20 years, since the early 1990s, we've actually seen a doubling in the number of people in Canada who are living with kidney failure. And you might ask yourself, well, how can you be living despite having kidneys that have failed? And the reason is really one of two treatments. Either those patients are on dialysis, which means we're cleaning their blood artificially with a machine, or they've had a kidney transplant, where we've taken a kidney from one person that has working kidneys and put it into the other person to replace the function of their failed kidneys. And interestingly, if you ask people who've had kidney transplants, which would you prefer, dialysis or transplant, especially people who've done both, almost everybody tells you they prefer transplant. And there's a number of reasons, but some of them are that they tend to feel better, so their quality of life is improved. And they also tend to live longer. And of course, they don't need the machine hooked up to them 3 times a week to clean their blood, which can be quite cumbersome, as I think you can imagine. Also, though, from a healthcare perspective, kidney transplantation is actually significantly cheaper than dialysis for the taxpayer. And so it's really a win-win situation, and we would actually do many more kidney transplants if we had the organs available. But that's the one main limiting factor. So if any of you have ever known somebody who's had a kidney transplant, one of the things they'll often talk about is how well they matched with the kidney that was donated to them. And that's my specific area of interest and what I'm going to talk to you a little bit about today. I think many of you probably understand the concept of matching already, because I bet if I wandered through the tables and asked you guys, many of you could probably tell me what your blood type is. And the reason that we know that information, and it's been drilled into us over the years, is because if you need a transfusion tomorrow, it's very, very important you get the right type of blood. It's critical, in fact. If you're an A type and you get the wrong type of blood transfused into you, that will actually be immediately recognized by the immune system as like a foreign substance entering your body, and it will attack those cells and destroy them in a matter of minutes. And in fact, the response is so vigorous it can actually make you worse than you were before and even lead to death. So in hospital, we're very careful about giving you the right blood, as you probably know. And in fact, transplantation is no different. If you get an organ from someone else, when that organ comes into your body, the immune system says, oh, that's something foreign. We need to attack and destroy. And unfortunately, unlike blood typing, where there's only actually 4 blood types to worry about, in transplant we worry about the 4 blood types, but we also have this other group of proteins, which are a small group of proteins, but there's thousands and thousands of variations. And we are trying to match for all of those variations in those proteins. So it's actually very unlikely that you get a transplant that's a perfect match. And as a consequence of that, almost everybody who gets a transplant of any type, solid organ anyway, needs to be on medications to suppress their immune system for the rest of their lives in order to prevent that attack that I was talking about. Now, to give you a bit more detail about my research, what we're actually matching for are proteins. So these proteins are on the cell surface. And these proteins are made up of building blocks. You can kind of imagine them like Lego blocks. And if I was to dump a big pile of Lego in the room and ask you guys to all build me a Lego house, what we'd end up with is a huge variety of different houses. And these proteins on the cell are like that. There's lots of variety, there's lots of detail, and if we take a look at them carefully, just like if we took a look at these houses carefully, we could start putting them in categories, like, for example, duplexes, or 2 stories, or bungalows. And these proteins are kind of like that. Historically, what we've done is we've looked at these proteins and we knew that there was a lot of detail there, but we didn't have the tools necessary to see it. And so we took what we could see and we grouped them. And then we took people that had bungalows and we matched them with other people that had bungalows. And for the most part, that's served us fairly well over the last 20 or 30 years of transplant. But recently, the technology's really advanced so that now we can see these proteins in much, much greater detail than ever before. To take the Lego analogy, instead of just matching bungalow for bungalow, we're now matching bungalows with green doors and blue windows and pink chimneys to kidneys that have those exact same characteristics. Furthermore, in my research, we're very interested on looking at the proteins themselves to see if there's areas within those proteins that are more important to match than others. So to bring you back to the house analogy, imagine that you have a kidney with a bungalow on it, and your bungalow has a pink chimney. What we're asking is it very important then that you also get a kidney that has a pink chimney on it? And in fact, we're finding that's to be the case. In fact, it's much more important to match, say, the chimney than either the doors or the windows or the eave troughs or things like that. So you might wonder, well, how can we use that type of information? The main way that we use this or want to use it is to try to avoid, first identify, and then avoid these very high-risk mismatches at the time of transplant, so that the kidneys that we do transplant can last a long time. The second way is, as you can probably imagine, you can develop some sort of a risk score based on how well these kidneys are matching. So for example, if we have a patient who receives a very high-risk mismatch, which sometimes occurs because patients will take sometimes whatever kidney they can get, those people will need a higher level of drugs to bring the immune system down or suppress it. Whereas if you have a person over here with a low-risk mismatch, you can maybe get away with a little bit lower immunosuppressive medications. And so in this way, what we're trying to do is personalize or individualize the medicine that we need to give people, instead of using kind of a one-size-fits-all approach. So this is one of the ways we're using this, and there are other ways as well, but I'll leave you to think about that, and hopefully it'll bring up some questions, which I'd be happy to address once all of our speakers have had a chance to talk. Thank you.
Thanks, Chris. Okay, our next presenter will be Samantha Pauls. Samantha obtained her Bachelor Honors degree from the University of Manitoba in Biochemistry is currently pursuing her Ph.D. degree in immunology. And her research, as she's going to tell you about, focuses on signaling networks in B lymphocytes.
Hi, so I'm here representing a more basic approach to research. And what that means is that I don't focus necessarily on one disease specifically, but hopefully my research is applicable broadly to several different disorders and conditions. So my work is in the field of something called cell signaling. And this is the study of how cells deal with cues that are found around them. So for example, this could be a virus particle or an allergen or something the cell needs to recognize and respond to. So what happens is these cues don't usually enter inside the cell. What usually happens is they just hit the surface. And on the surface are specific proteins that recognize these cues. These are called receptors. So it's the receptor's job to relay the signal inside the cell. And then from there, the signal can be passed from player to player to player, usually proteins, until finally the cell decides what to do with it. So a B cell, for example, might start dividing or start making antibodies. So cell signaling is kind of like a big relay race. So the receptors are the starting lines, and each player runs its part and passes the baton to the next. But now imagine a relay race where there are dozens of potential starting lines and dozens of potential finish lines. And imagine that you could share teammates between teams and that there are alternate routes you can take. Some teams help other teams out and help them go faster, while other teams actually sabotage their rivals. One more step further, imagine now this is all happening at a microscopic scale, and you can't actually see any of these players directly. So how would you ever figure out what was going on? How would you figure out the routes that are taken and where they lead? Well, you would do so by experimentation and inference. You would mess with one player and see what happens to the players around it. So this is essentially what we do as a signaling research. This is how we build maps of the signaling pathways and see what they mean. So we've mapped out a lot more than we had a decade ago, but there's still many, many gaps in our understanding. So why do we care? Well, we care because many diseases, for example, autoimmune diseases, cancers, and allergic diseases, they can arise because of defects in cell signaling. So you can have false starts at the starting line, you can have rogue players that just run their part without waiting for the baton, you can have players that get tired and just can't go any further. And so a way to treat these diseases is to target these defective players. Either fix them up or bypass them and somehow manage to restore order to the system. So it's really important to do research in cell signaling because first you can identify new therapeutic targets based on these defective players, and also it just helps us build a more accurate picture of what's really going on so that you can start making predictions and predicting problems with existing therapies, kind of based on the players that are connected to the target you're after. So what do I do exactly? Well, I research a specific protein. It's called SHIP, or SHIP. And this is a signaling protein that is involved in a cell signaling network. And it's actually one of those signaling proteins that sabotages its own team. But it's for a good cause, because it prevents the cells from reacting too strongly to cues in the environment. So, proteins get modified. The best example of this is a chemical group that's added to it, and this changes how the protein works. Kind of like how a new pair of runners might make you run faster. So the thing is, there is this specific modification that happens to SHIP, but we have no idea what its purpose is. So it's my job to figure that out. So the way I approach this is I force cells, or I grow cells in the lab, and I force them to make either regular SHIP, or I force them to make a version of SHIP that can no longer get modified. We call this a mutant version. And then I see what changes in the cell's behavior and what changes in the signaling network of the proteins connected to SHIP. And this way I can figure out what the purpose of that modification is. So why do we care? Well, again, we're filling in a very important piece of that map. Also, this site could be a potentially new therapeutic target once we understand what it does. And finally, there's already drugs in clinical development that target a different part of this same molecule SHIP. So it becomes very important to figure out really what functions of SHIP are attributed to which site so that we can predict when these therapies will be effective or when they will actually be useless or even harmful. Thank you.
Okay, speaker number 3 is Natascha Fitch. Natascha obtained her bachelor's degree in biochemistry also from the University of New Brunswick and is currently pursuing a master's degree in immunology here. And she's working on understanding the effects, specific measurable effects of vitamin D on immune cell function.
Thank you. So I'm going to start off with a little bit of a self-introduction, a little bit more of how I got into immunology and research in general. So when I was younger, I went to science camp, which was where I first learned my first set of kind of experiments and got introduced to science and what it really meant. And from there, I always liked science. However, I wasn't really introduced into immunology until I went and did my undergraduate degree where I did my first course. And what I found out was that a lot of infections and diseases actually relay back to the immune system and to errors in the immune system. And so it plays a huge role in basically any sort of these health issues. So that's how I really got interested in that. And so from there on, I decided to do my master's in immunology. And today I'm going to talk to you about my project on vitamin D. So all of you may have heard about vitamin D somewhere in the media, and if you have, it's probably you've heard about its role in bone development and how it's very important to keep strong and healthy bones. But actually, vitamin D is a hormone, not a vitamin, whose precursor is located in the vitamin D pathway, and then it gets transported to other areas of your body, organs, tissues, cells, and then the cells will then modify this version of vitamin D, and the active form gets used by the cells to do a lot of different functions, and one is actually to regulate a lot of the immune system. So that's kind of what my project more involves. So vitamin D can also be found in the diet, however, mostly we get it from the sun. It can be found in cod liver oil as well as other fish byproducts, and there's lots of supplements and things that are now put into milk and other products like that. So maybe you've heard of vitamin D sufficiency and you're not quite sure what that is, or maybe you know exactly what that is, but basically what that refers to is it refers to the circulating levels of vitamin D within your system that are above a certain level that doctors and researchers have deemed that are appropriate to help regulate these responses. So if you're vitamin D sufficient, you have enough vitamin D in your system. However, if you're insufficient, you're just below that, and if you're deficient, then you're really lacking vitamin D. So why should you care about being vitamin D sufficient? Well, actually, low vitamin D status is widespread amongst Canadians, and some of the research that I've done recently actually has shown shockingly low levels amongst adolescents here right in Winnipeg. So I have a cohort of people who I've analyzed here, and a lot of people are very deficient in vitamin D, and part of that is just because of the location. We don't get a lot of sunlight, and the sunlight that we do get doesn't have a whole lot of UV rays that are needed to break that precursor. So vitamin D, although originally known for its role in bone development, like I said before, also plays a large role in regulating the immune system. And vitamin D deficiency has been linked to several autoimmune diseases, including Crohn's disease, as well as diabetes. And vitamin D deficiency as well has been linked to increased upper respiratory tract infections, so that includes influenza, as well as other viral infections. However, to date, this is mostly statistics that have shown this. We haven't been able to really shown it in the lab yet. And so there is also speculation that vitamin D levels may have benefits in preventing the pathogenesis or exacerbation of asthma, so that's another reason why it's important to study. And so this kind of has led to my lab being interested in how vitamin D can actually regulate or modulate the immune system. So my specific project focuses more on the question, can vitamin D regulate the immune response to viral infections? And so to do this, I get blood samples from adults, neonates, and even children, and I separate their white blood cells from the rest of their blood. Then what I do is I take these cells and I add viruses as well as vitamin D, so I've done different viruses, and then I culture these cells or put them in an incubator overnight, and I measure the cell responses the next day to see if there's differences with and without vitamin D, or when there's viruses and vitamin D under all these different conditions. And so what I do is I measure how the cells respond, and to do that I measure something called cytokine production. And cytokines are a hormone that are released by the cell, and really what they do is they communicate with the other cells around them to kind of let them know what kind of environment they are. So if there's a bacteria and your cell recognizes a bacteria, it'll produce these cytokines to kind of let the other cells know what's going on and saying, hey, I need some help here. And so to date I've found that vitamin D can modulate the immune response to viral infections. However, I found that this is largely dependent on the type of virus that is introduced to the cell, and as well as the type of cell that's examined. So there's many different types of cells in your body, and depending on what type of cell you look at, vitamin D can have different effects on that cell. So how will studying this help healthcare? Well, my research specifically is important because it helps us understand the potential for vitamin D to aid in the prevention and the treatment of viral infections. So viral infections are very prevalent amongst the population and they're hard to treat. So having a way to prevent these infections or just lowering the amount of people that are infected, get infections is very important. And consequently, my research will also help to target people who are at risk of developing vitamin D deficiency, and also target those individuals who are most likely to benefit from vitamin D supplementation.
Thanks, Natascha. How's everyone doing out there? More than halfway through? You can hold your burning questions for 2 more talks, and then we'll have a great discussion, I hope. All right. Next speaker is Samantha Pauls. He obtained his bachelor's degree in microbiology from the University of Tehran and a master's in medical microbiology from the Pasteur Institute of Iran, and is now pursuing a Ph.D. degree here in Wiebe. And he's going to tell us about his research on recruitment of immune cells to inflamed lungs and diseases like asthma.
Thank you, Dr. Marshall. To start my talk, may I ask all audience in this room to take a deep breath and think about how fortunate we are that we have no serious issue with our respiration. Despite remarkable achievements of immunological interventions such as vaccination in controlling communicable disease like infectious diseases, there is a huge unmanageable increase in prevalence of allergic disease along with urbanization during the last decades. For instance, asthma emerged as a serious public health issue. Medical doctors define asthma as a chronic inflammatory disease of the airways which causes tightness in the chest and shortness of breath. The most common type of asthma is allergic asthma, which is the focus of my studies here as a PhD student. It triggers by exposure to some harmless substances called allergens, which induces recruitment of some inflammatory cells to the airways, and it makes the airways as a battlefield to eliminate those allergens. Over time, the airways become inflamed and sensitive. If this airway inflammation is not diagnosed properly and consequently if it's not treated well, it will lead to a fatal asthma attack. Asthma is the leading cause of absenteeism from the school and the third leading cause of work loss in Canada. 13% of Canadian children and more than 8% of Canadian adults have been diagnosed with asthma. And unfortunately, Manitoba has the highest prevalence of asthma in the country. Unfortunately, there is no specific therapy for asthma, and current anti-inflammatory drugs, which are not specific for asthma, cannot cure severe cases of the disease. And again, unfortunately, it's associated with several side effects, which we cannot do anything with them clinically. So there is an unmet need to develop novel strategies for treating asthma as well as its diagnosis and prevention. And it definitely needs to obtain more immunological knowledge about the cellular and molecular mechanism underlying asthma pathology. As a PhD student of immunology, in collaboration with other groups inside and outside the University of Manitoba, I intend to broaden the horizons of knowledge about immunological aspects of asthma. During the last 3 years, I have discovered a molecule which is called semaphorin 3E. The amount of this protein is much lower in asthmatic patients compared to healthy individuals, and it may provide us with an innovative advancement to diagnose asthma in more efficient way compared to current diagnostic approaches. I have expanded also my study using an animal model of asthma. In this model, we challenge mice with house dust mite as a typical allergen, and then we evaluate the effects of this molecule, semaphorin 3E, on asthma, on different aspects of asthma pathology. The thing that we found was quite interesting. We surprisingly observed that treating mice with our molecule of interest decreases airway inflammation and also improves lung function. We further characterized detailed nature of inflammatory responses in the airways of asthmatic mice, which semaphorin 3E has been genetically knocked out in our model. In conclusion, our immunological research may open a new avenue to better control asthma in terms of its diagnosis, treatment, and also prevention. However, it definitely deserves more study to address all concerns regarding the efficacy, the mechanism of action, and also safety of this approach. Thank you.
Last presenter is Rebecca Dielschneider. Rebecca obtained her bachelor's degree in microbiology and immunology from Dalhousie University in Halifax, and is currently pursuing her master's degree here in Winnipeg in immunology. And her research is looking at disruptions of those signaling networks that we heard about earlier, and how they can cause immune cells to turn cancerous and become leukemia or lymphoma.
So as Dr. Marshall said, my name is Rebecca, and I study cancers of the immune system. And from a scientific standpoint, I find cancers of the immune system to be particularly interesting because the cells in your body, your immune cells, which are supposed to detect cancer and protect you from cancer, actually go rogue and become cancerous themselves. The particular leukemia I study is called chronic lymphocytic leukemia, and it is the most common adult leukemia in North America. And although many treatments and many therapeutic options exist, the toxicities can be quite severe, and unfortunately relapse or recurrence of the disease is quite common. So the lab where I work, we're focused to finding new therapeutic options that are less targeted, or less toxic, more targeted, and that will hopefully really eliminate the cancer cells and leave your normal healthy immune cells. And you can consider a normal cell kind of like a construction company. And this construction company builds buildings at a normal rate to sustain an active city. And it really takes the time to build quality buildings that are well supported. In contrast, a cancer cell is like a construction company that builds at an abnormally vigorous rate, and it can overwhelm a city. And despite the buildings may look strong and may look normal, this company is actually cutting a lot of corners, and therefore the buildings lack some critical support and lack quality and integrity. So because this company has had all of this advantageous gain, and it's growing so fast and building so fast, it can actually be quite vulnerable. Using that analogy, it can actually be more vulnerable to inspections by city officials, like our immune cells. And its buildings, because they lack some of that quality, can actually be more vulnerable to damage, like cancer cells are to certain therapies. We often think that cancer cells are so strong and resilient, and in some ways they are, but really each strength can also be considered a weakness and a vulnerability that we can target in therapy. And in the lab, I test the various weaknesses and vulnerabilities of leukemic cells in the blood donated by leukemic patients at Cancer Care Manitoba. And in the lab, I process blood samples and I isolate the cancer cells, and then I can add different drugs and different stimuli and different cues like Sam was talking about, and then later measure different cellular responses and different effects. And in some of my experiments, I've actually found that one drug can particularly target a subset of cancer cells that are the most aggressive type, and not target non-aggressive cells, not our normal cells. And also I found that a different drug can actually target a specific vulnerability of drug-resistant leukemic cells, which are very prevalent, but not actually target our normal immune cells, which is of course a benefit to a patient. And targeted therapies like these are really needed to develop medicine that is personalized not only for different types of cancer, but also just for different individuals. Just as each person is different and just as each person is an individual, so is their cancer. Even if they all have the same type of leukemia, their leukemia can still all be very, very different. And so therefore multiple different therapeutic options are needed to target the vulnerabilities of their specific cancer cells. And really we can't do this without research. Research is really what teaches us about the unique aspects of cancer cells and what makes them stand out compared to our normal cells. And research enables us to test different drugs that we hope will someday be used in the clinic to target cancer-specific vulnerabilities. Thank you.
Thanks, Rebecca, and thanks to all the panelists for the brief summaries, and everyone kept on time. And I'll just say before I give you guys a chance to ask questions that it really strikes me hearing these guys talk. This is why I like immunology, is the real variety. We have personalities, but we have very clinical, very early stage research, very advanced stage research, many different health issues where there's immunological underpinnings, and I think that's if it seems like a diverse set of talks, that's the common theme that we want you to be able to see from this set of presentations. Okay, question time. Just, do be patient with me. I'll try to get to everyone with a question and start with Tim. Thank you. I'll come to you.
I wanted to know if aging and immunology are being looked at the university.
Anyone want to have a shot at that? I can
talk about that a little bit.
Yes, Chris.
One of the things that I did on a previous research project was look at risk factors for patients who develop antibodies in transplantation. And so we looked at a whole bunch of clinical risk factors, everything from the medications they take, to the age, to the different races that we have in our population, a whole variety of things. And one of the things that actually pops out was age. In fact, for those of you who are statistics oriented, there's ways that we can adjust for differences between patients. It's called a regression model. I won't go into the details, but even after adjusting for all the factors that we could, age was still an independent risk factor for developing antibodies. It's not older age, it's younger age. So as we think that in this model, at least of transplant, those who are youngest, and by youngest I mean teenage years, we're not transplanting people in their infancy, where your immune system is a bit weaker, but people who are younger in our cohort of patients had the most robust immune response to transplant, and they therefore were the most likely to form antibodies against the kidney that they received. But as the ages went up, the chances of that happening went down. As you can probably guess, there's lots of other possible explanations for this other than just a robust immune system. For one thing, younger patients don't tend to take their medications very well. We try to adjust for some of those things, but it's never perfect. It's based on what they tell us. So that might give you a partial answer to your question.
Rebecca, you want to say anything about CLL and age? Or I'm happy to.
Well, I mean, the leukemia I study is the leukemia of adults. So as we all know, our immune system does get weaker as we age. So that is something that we try to take into consideration when we're looking at new therapies, that we are targeting the immune system that is potentially already a little bit weak.
And I'll say that there is a lot of studies done in that area, but there's very, very clear changes, age-related changes in your immune system that we don't really understand. So you can measure a lot of changes, but it's like the dinosaur bone. So we have a pile of dinosaur bone, but we can't really put it together to say, here's the big picture of the difference between the immune system at different stages of life. Other?
Sorry, I also had a question for speaker number 3. I'm sorry, I couldn't hear your name.
Natascha, yeah.
In the a newspaper article lately, they've been mentioning vitamin D and multiple sclerosis, particularly Manitoba is a hotbed of it. And when you mention vitamin D, are you looking at that at all?
No, actually I'm not. There's a lot of study that's going on all over the place on different diseases and how, taking vitamin D supplements can alter your immune response and sort of help improve the progression of certain diseases. So multiple sclerosis is definitely one of them. I've read some papers on there, but my project doesn't really specifically focus on that. Later on, I'm more focused in viral infections and actually in asthma and allergies. So there's a certain virus called respiratory syncytial virus, which really affects individuals with asthma, and that's kind of more where my project is headed towards, not really multiple sclerosis. But yeah, there's lots of interesting things out there.
Also, too, feel free to ask something in response to something that somebody said, either in the audience or in the place.
I haven't heard the word glutathione mentioned at all. Anybody talking about it, researching it, working with it?
Sorry. Earlier this month, I actually went to a really big cancer conference in Washington, and cancer metabolism is a huge field, and so many people are talking about it there. Specifically, I don't study it all that often, at all. I certainly read about it, and just the metabolism in general of cancer cells is really altered and really interesting to study. If you think about it, because the cancer cell is trying to divide much more rapidly than it's supposed to, than it's programmed to, it needs to increase its metabolic rate accordingly. And glutathione plays a role in that, but I certainly don't know a lot of details, sorry.
Thanks.
Are you aware that in the Canadian Pharmaceutical Compendium there is a product which specifically raises the immune system and naturally forming glutathione? Like glutathione has to be made by the body, and there's 4 pages in there that talk about that. Are you aware of that information?
I'll take that one. There are many such observations of natural and foreign plant, animal nutrients that can boost your immune system in various ways. And it's just, vitamin D is in that sort of category, vitamin C. And it's complicated. That's the sort of short answer to the question, is you can observe, so there's observations are all out there for many, many things, but there's one can't come up with a sort of pat answer to say this is the cure to X, Y, or this is an immune booster. That's actually sort of a term to teach medical students occasionally, and I always encourage them to Google immune system. The first 10 hits that come up aren't science at all. They're sort of products, immunoboosts. But the thing is, there's actually evidence often supporting a lot of those things. So I'm not aware of those 4 pages of information on this glutathione specifically, but I'm aware that a other examples like that. So that's the answer from my point of view. Does anyone have any additional knowledge of that in particular?
What's a question? It's a statement.
Right, yeah, so we're actually looking for
What
questions.
Maria is saying is that in the CPS is protocol and tests, testing will be removed.
Yeah, sorry, yeah, we're looking for questions actually, not statements. Any other questions?
I wondered, when it comes to organ donations, as we age, does there come a time when you should not bother to donate your organs anymore because they're no longer useful?
No, in fact, we don't even have a hard cutoff for, in terms of an age where you can't receive a donation either. But it basically comes down to what the kidney function is at the time that it's available. So there isn't, if you have a perfectly functioning kidney, you take it just about any age. As you can probably guess, we are selective in terms of, we want to only take kidneys from people who are very healthy, because we want to make sure that the one kidney they have left is going to be with them for the rest of their lives and work well for the rest of their lives. And so if there's other complicating factors like high blood pressure or diabetes, we won't take those. Does that make sense?
Thanks. Question? Through the chairs now.
Well done. I got a question, I guess, regarding gene therapy in amongst all this, because I'm a bit of a dreamer, and well, when the first human trials begin to sign me up. But what role does that play in all this? I know you guys are definitely in a role in prevention, at least finding, attacking the cause of the source, regardless. What can be done? I guess is that a parallel realm to what you guys are doing, or is that an alternative avenue?
That's a great question.
What was the question?
I think I'll rephrase it. I think if it's fair, is gene therapy one way that we might target when we talk about targeting something that's gone wrong to fix it? Is gene therapy one possible way that might happen? And I'll say yes, it is. It's still being actively pursued. We don't have that student up here, but we have people in our Department of Immunology pursuing that specific approach. It's technically very challenging compared to the conventional medicines like enzyme inhibitors, which are a small molecule that you can take in a pill. Gene therapy is much more complex, but it may be the way to go in some cases.
I think in newer approaches, people are trying to combine gene therapy, which was a sort of traditional kind of approach, with cell therapy, especially in the case of immunological disorders, like getting benefits of both some cells which might be not working properly in different immunological diseases, and the genes which are highly expressed or their expression is decreased, doesn't matter. People are trying to combine these two approaches to have better, more efficient alternative approaches. It's not, gene therapy in most of immunological disorders is not effective by itself.
Does our immune system ever learn? Does it get better at certain tasks, or does it always get, as we age, get worse?
Great question. Great question. These are students. This is like a test, the final exam.
I can have a crack at that. So I don't know if you're familiar with the term immune memory, but so what happens is the first time you're exposed to an infectious agent of some sort, you'll have an immune response, and hopefully you will clear that infection and get healthy again. The next time you encounter that same pathogen, you'll have a better immune response, and you'll get better faster. And so the reason this happens is because during the first immune response, you actually have a few cells that stick around. We call them memory cells because they're long-lived. They are specific for that pathogen. This can be B cells or T cells usually, and they can wait around and just kind of troll the body until that same pathogen comes back and it attacks it right away. And so some immune memory cells can last years and years and years, and some are a little bit shorter lived. And so actually this is largely the basis for vaccinations. When you're vaccinated as a child, you're introduced to certain elements of the pathogen they're trying to protect you against, so that when you actually see the real pathogen, you will mount a strong immune response and clear it effectively. And so, yeah, I guess these cells can stick around while you age, and they diminish over time as well, but they certainly can stick around and help.
Excellent. I'll give that in, I don't know, B+.
I could add a little bit too, maybe. Bringing it up closer. Just in addition to what Sam was saying, there's often ways that we can actually teach your immune system something that perhaps it hasn't learned on its own. The fancy word for that is called immunotherapy. It's basically just teaching your immune system to target something specifically, and that's a common approach in cancer. Specifically, if the cancer cell is quite different than its normal counterparts, you can actually try to teach your immune cells to recognize that, and then they can do the rest, and they can actually eliminate the tumor, and then you don't really have to treat a patient with really damaging agents. And I know that that's really an approach that's tried to be used in many other diseases too.
Right, I'm going to add something on this because I think it's really fascinating. It relates to the aging in the immune system. Like, why shouldn't the immune system just get better and better and better, right? It's learning. It's had more experience with more different bugs. It should be like super ready. But I think one school of thought about this declining immune function over time is that almost the immune system gets set in its ways and it becomes, we call it oligoclonal, which means that your immune system, instead of being very open-minded and able to respond to thousands of things, now it's very focused on the sort of 5 or 6 things that have troubled you several times over in your life, and a very big part of the repertoire of your lymphocytes is dedicated to a few bugs. So it becomes sort of less flexible, less adaptable, but it has to do with your sort of evolutionary history of your immune cells interacting with the different infections that you've had over life. That's one school of thought, but again, we really don't completely understand what happens. Some people actually think CLL is an example of sort of one clone of lymphocytes that was once useful to you to defend you from an infection, but it just kept going and expanding until it filled up your whole entire immune system and more.
She could elaborate on how we teach the immune system to suppress these cells, so you don't have to use the drugs. Could you elaborate on that?
Sure. Actually, it's kind of in addition to what Sam was saying before in response to the gene therapy question. A lot of the immunotherapies are cell-based therapies. So you could actually take the immune cells out of a patient and actually expose them to a particular protein that's only particular to the cancer cell, and actually teach them in the lab to respond to that protein and then actually administer them back into the patient. Whereas in many cancers, the cancer is actually suppressing your immune system to the point that it's prohibiting your immune cells to do that within your body. So that's why we can take them actually out and teach them in the lab and then administer them back in. And there's many different cells that are involved in that. A lymphocyte you can take out and manipulate like that is called a T cell. There's also many dendritic cell-based therapies. And a dendritic cell is a cell that will actually help to teach your T cell and to help a lot of your immune cells, and it is an immune cell in itself as well. So does that answer your question? I didn't go too much into details there, but it's certainly an interesting field of research, and it's not been around for too long. So I think it's...
Is that something like years ago we used to have these autogenous vaccines? Is that something that...
Sorry, I didn't hear.
Yeah, I didn't quite catch it.
Well, years and years ago, they used to have what they called autogenous vaccines. Is that something similar to that?
I think so, yeah.
They would actually take patients only.
Maybe a comment from another regarding your question.
Yeah, lately this technique has also been implicated in asthma. that very high doses of allergen are given to these asthmatics, so that when they get those allergens which are everywhere around you in the form of pollens or dust, so these people, they are not responsive to them. So this immunotherapy is also used in asthma. Secondly, this very phenomena also lays a foundation of a term in immunology, which is hygiene hypothesis, which means people who are exposed to more bugs, they are more stronger to fight against them. So that shows that they're learning during the course of exposure, and their immune system is like completely taught that how to fight against these bugs.
Thanks. Have a question? Sorry, she's also a student in immunology. She's
I just wanted to know if you guys are coming up with any more research for people that have immunodeficiency disorders.
In our particular department, so in the field of immunology, lots. I mean, that's a big area. We don't have a particular big presence in that. It's a big, huge area of immunology. We don't have a huge presence there. But that's one area, for example. There's genetically determined immunodeficiencies where gene therapy is a big player. But yeah, if you're talking about locally based research, I can't tell you a nice story about that one. But that's, yeah. I think a lot of the research that's happened over the last, in my career in immunology, is really identifying the genetic basis for these immunodeficiencies. And once you know what's wrong, that gives you something, but it's still 20 years down the road before people figure out how to correct that. That's a lot of it. Spot back here.
Natascha, I think you said about the vitamin D deficiency, that it was much more widespread. Mm-hmm. My question is this usually tested for, like when you go for a CPC or whatever, and because, well, you don't want personal stories. Anyway, the way I thought about it was a special test that was done. But how would you know that you have a vitamin D deficiency if they don't sort of regularly test for it?
Right.
If it's so widespread here, it seems to me maybe a good idea to test.
Exactly, yeah. So from what I know, it's not like routine to get vitamin D testing done when you go to the doctor or anything like that. For me, for my research, I have a cohort of individuals, so a group of individuals who come in, and I actually send away their plasma samples, and plasma is just a layer in your blood. Once you spin it down, you get rid of the cells and stuff, it's just the proteins, and then I send those samples away and they get measured for vitamin D so that I know what the levels are of those individual cells who I'm working with. Then I can make correlations and kind of make connections that way. But mostly I think that if you go to the doctor and you do get vitamin D testing, it's more if you have something like Crohn's disease or diabetes, where it's known that a lot of the population will be, or a lot of those people will be deficient in vitamin D, and that they're requiring supplementation. But most of the time they will just give you or tell you to take vitamin D if they think that you are deficient or that you could be deficient in vitamin D. No. To be honest, I have no idea how much it costs, but I can assume that probably yes.
That's you have really a really big. That's links to a larger issue, which is the more we know, more biomarkers. So we would call these things biomarkers, something you can measure in a lab that tells somebody about their health status that can inform the clinical practice or the recommendation to the patient. The number of possible things you can test for is just because we're learning more and more and more, and then guys like Chris have to tell us which ones are the most predictive, have the most predictive value to really tell us something that's important. That this is telling us more than the other 10 things, related things. And these are the ones we have to routinely test for. These are the ones we have to test for when there is the symptom X, Y, and Z. Then we move to that test. as like a healthcare management.
Yeah, I can understand what you're saying, but if the move is to preventative healthcare and that we're responsible for our health, if we have to test for things that might bring on these symptoms, another chronic illness or something, it seems to me somewhere we have to figure that out.
Yeah, no, definitely. It's a huge issue.
Yeah.
Just go to the cancer conferences where there's 10,000 biomarkers we could test for, which ones should we, and how do we interpret the data afterwards.
It takes months for that test result to come.
Pardon me?
It takes months for that test result to come.
Natascha, can you explain the difference between oral vitamin D and D3, please?
Yes. Okay, so the vitamin D that you take like a supplement, it's not actually the active form of vitamin D that's used by your cells. You can't actually just administer that yourself. So what will happen is if you take an oral supplement, it will go through several rounds of metabolism before it becomes this active form that your cells or your kidneys can actually use. So it becomes a little bit complicated because you can take these supplements and it can raise your circulating vitamin D levels, but whether or not that's actually doing anything in your body, it's very difficult to say because the active form of vitamin D is really only found in tissues and in areas where it's used. It's not really found circulating, so that comes back to a different point with that, which is actually looking at circulating levels of vitamin D. They're not very accurate as to whether or not vitamin D is being used. So when you're taking an oral supplement, it still needs to go through several rounds of modification in your body before it becomes an active form that can be used.
I can maybe add something to that end. If you're listening closely to Natascha during your talk, you will have heard that the kidney is one of the last steps in vitamin D production. Skin is the first step, kidney is the last step. And so I'll answer a few questions here maybe at once. In our clinic, we don't measure almost nobody's vitamin D level, and the reason is we know that everybody's deficient, because in my world, everybody has kidney disease, and so none of them do this last step very well, which is the production of the final step is called calcitriol. And so we actually give everybody in our clinic vitamin D3 or calcitriol, because we know that they can't do that last step. To expand a little bit on your other question, I'm trying to stay quiet, it's not my area, but we have many biomarkers that we can test, but for the vitamin D, we know that people who have bone disease or malabsorption syndrome, that they're going to likely be deficient, or that fixing the problem will make a big difference for those people that have bone disease. For these other things, we don't know yet, and so to test for something that we don't know will make a difference is a slippery slope, and I'll go back to the MS example that somebody mentioned before, where a test gets identified that may or may not have a benefit. Suddenly thousands of people are going to have their brains checked to see if they have a narrowing of blood vessels without any real good proof that that means something. And then people are having a test that has a lot of potential harm, even though we don't know that it has any potential good. And so we're very careful in medicine about recommending population-wide tests until we know for sure that it's going to make a difference. Vitamin D is not quite the same example because, like somebody said, if you really think you're deficient, it's very safe to just take some vitamin D. That's why we give it just what everybody wants.
So my question about taking vitamin D, is there an overdose level?
Yeah, so a lot of people have sort of been concerned with toxicity levels of vitamin D, but actually to date there's not really any certain level that's considered toxic, because you actually do have an enzyme in your cells that will convert the active form of vitamin D to inactive if you have too much. So vitamin D has a way of regulating itself. So yeah, I mean, like Chris was saying, if you're deficient in vitamin D, or if you think you might be, it's safe enough just to take supplements, because it's very difficult to overdose yourself or to have too much vitamin D.
I'll add I'm not sure what I'm looking at, forget to ask the question. one more thing, I'll just say that it is possible to overdose in vitamin D, and sometimes your body can actually accidentally make too much vitamin D. In fact, it's a very well understood complication of some cancers, in particular lung cancers, that actually produce too much vitamin D. And what the end result is not that you're going to die from vitamin D, but you end up, the whole purpose of vitamin D is to absorb calcium from your gut. It tells your intestines when to absorb more calcium. And so you can actually get such high calcium levels that you get symptomatic from that, and that can produce muscle spasm or tetany and other confusion, constipation, all kinds of things can happen if you get very high calcium levels.
More vitamin D. When you do a kidney transplant, you suppress the immune system.
Yes, we do.
And you give them vitamin D. You said everybody in your clinic gets vitamin D.
Yeah, that's a good point. So I'm talking about the chronic kidney disease clinic. We have a very interesting thing that happens in kidney transplant, because we take patients who are on dialysis, and probably the average dialysis patient's on about 10 or 12 medications, and we can stop about half of those the day that we do the kidney transplant, because we can restore all of those functions back that their kidneys they were born with were doing for them in the first place. Unfortunately, not every kidney transplant results in 100% function back. So in many cases, we get some function back, but not all of it. So it depends how much function, whether or not they all still need vitamin D in the transplant clinic. So in the chronic kidney disease clinic, pretty much everybody needs it. In the transplant clinic, I couldn't tell you a percentage, but it depends where you are after transplant. This is not a curing therapy. Even the transplants gradually fail over time.
I can just add to that too, so we're just kind of going back and forth here, but so vitamin D, it can decrease or suppress your immune system in some ways, but it has also been shown to increase something called antimicrobial peptides, which are basically these peptides that can fight off bacteria and directly kill bacteria. So although it can suppress your immune system in one sense, it can actually heighten it in a different sense. So it's more of a regulatory vitamin, more so than even just like an immunosuppressant.
Okay, lots of interest in vitamin D. Any leukemia, asthma, autoimmunity, signaling networks and lymphocytes, that's my favorite part.
Just wondering, back to the I guess the saturation of allergens regarding asthma has been commented. Is there any merit to that? I know you can definitely. I know.
That's it.
The important thing for your question is that the nature of allergen is important first of all, over the saturation step or whatever it is, both clinically and also in research level, both in human and mouse. We need to know what we are specifically sensitive or allergic, then think about in which level we are sensitive to that allergen to develop our therapeutic strategies or whatever which helps control the disease. But the fact that asthma is growing along with urbanization tells us that all these allergic and even non-allergic triggers of asthma are always in our environment, especially in industrial countries like Canada. Back to the point about hygiene hypothesis, which somehow supports this idea. So the thing here is that we cannot get rid of the presence of allergens based on the current lifestyle in industrialized countries. The thing that we can do is at the research level to think about some other alternative, as I said, some new innovative approaches to elaborate other arms of immune system to fight with presence of allergens. We cannot do anything with allergens in terms of their presence or amount of it or something like that.
I'm just going to throw this out there, and I'm not sure if everyone understands the idea about allergen immunotherapy or this high dose tolerance, or what was the word you used? Saturation therapy, because it's absolutely counterintuitive, and it gives insight into what we deal with as immunologists. So the way to turn on your immune response against a virus is give you a dead virus, give you an immunization, your body makes a response. The way to turn off an allergic response against an allergen is give you the allergen, lots of it, over and over again, get your needles, right? The fact is we don't really well understand why that works. The immune system has ways of turning on in response to a specific substance and a way of turning off in response to a specific substance, and we don't completely understand all those things. But this allergen immunotherapy has been around, giving massive doses of Ragweed to people, and in a few weeks later they become less allergic. It's a few months later, and this has been known, it's a phenomenon, and it actually does work to some extent.
The unfortunate, sorry to interrupt, but the unfortunate fact about this approach is that many clinical trials actually fail in step 2 or 3, and they are not effective in human. If it was, people would continue to treat it because it's a traditional way of doing it, but it's not as potential as people would expect before.
Can I also add something? So, when we are talking about asthma, it's not just a disease, but we need to understand it's a hypersensitivity of a person to something. So, suppose it's a pollen grain, I am not sensitive to it, someone else is. So, it's not just allergen we need to target, but there are many things which are personal. So, it can be my genes, it can be something related to my genes and my proteins which are mine, has nothing to do with other people in my family, or may have to do something with my family members, something genetic, hereditary. So targeting allergens, giving high dose of allergens is just one factor we are trying to look at to treat asthma. But again, it's a question of sensitivity of a person rather than it's a disease. And it's a gradual process. It doesn't take place just in a month or a week. So I hope that way we can have an insight that where this high dose or saturation dose or immunotherapy comes into play. So we are looking at many things, and we are just trying to find a treatment by targeting all these pathways.
Young children, especially preschool children who are going to preschool, seem to be sick all the time. Does this have something to do with the development of their immunology?
Well, it probably has more to do with their habits.
That's very tactful.
But you get sick because you come into contact with someone else who's sick or something that someone has left behind. So I think that's still the most, widely accepted reason that kids get sick. But there might be other things at play as well that we don't know about. It's really hard to tease those things apart when kids don't make good control groups because they're not careful with their hands.
So because it's come up with 2 or 3 times now, this hygiene hypothesis, again, people might not know what we mean. So one school of thought that's out there is that's kids being sick all the time as soon as they hit school age and are mixing around in big groups of kids who are all slobbering all over each other and all that stuff that they do. That's the immune system getting educated. One view is that that's good. That's exactly what kids need to do. They need to get sick. Their immune system needs to learn that we're not born with a perfectly functional immune system, that there's this education period that involves germs that we need to be exposed to. So this is what one had said about a hygiene hypothesis, is there's such a thing as too good of hygiene because we need exposure to germs. And maybe you've heard this, it's been out in the media a fair bit too. So there's actually some pretty good evidence for it. I'll give one little anecdote because it's amusing. One of them is that only children who are different immunologically. Children who they can control this based on number of siblings or whether a kid goes to daycare, whether they're exposed to farm animals, and things like this. There are lots of epidemiological studies showing that your exposure to germs from other children or from animals affects the development of your immune system. Not completely understood, but it's a fascinating idea.
I'd like to comment to that. So then, grandparents' immune system has to relearn so that we don't get sick from the kids?
I can make a comment. My wife worked in daycare for 10 years. She never gets sick.
Is the immune system your body's own antioxidant? Is it also the body's own detoxification system?
That's a good question.
Well, really the real detoxification system is the liver. As you may know, the kidney is really a filter that can filter out many of the things that have already been metabolized in the liver. So the liver is really a chemical factory that metabolizes the food that we eat, the drugs that we take, many things that we put into our body. It goes to the intestine. The first place it goes from the intestine is the liver, where many of these chemical reactions happen. So I think that would be the most accurate if you're looking for one place in the body. The kidneys would probably be number 2, although I'm a kidney doctor, so it's usually at the top of my list, but in this case, number 2. Immune system is more of a search and destroy type of end of things. If cells are damaged by oxidative stress, the immune system can see that damage, gets turned on, takes care of those cells. But I wouldn't say that they're the first players, so to speak.
Yeah, I think that is a fair assessment of general scientific opinion. And I think basically we as immunologists spend a lot of time learning about how immune cells, as a discrete entity, a cell, with proteins on its cell surface, how it recognizes threats that turn it on and make responses. And those threats generally fall into molecules associated with viruses, bacteria, and various things that infect us. So the way those cells respond. So the idea of a toxin, you know, is a large category. I mean, there's probably some things that people would refer to as a toxin that there's a receptor for on immune cells and they can respond to that. But in general, all the ways that we know that turn on your immune system and mobilize it to action involve microbes.
Can I elaborate on that?
I think we better give others.
Can I ask a question? Thanks. First, I'll congratulate all the grad students. Excellent job. Yeah, sure. Big applause for the grad students. Question is for Rebecca. You brought up this really interesting concept of targeted therapy in cancer. Now, we are in this era of antibiotic resistance for infections. Do you envision going down the road that cancer cells might somehow become resistant to these targeted therapies you're talking about?
I think we're starting to see that even now. Unfortunately, cancer cells are crafty, and a common theme in cancer cells is that they adapt and they can mutate, and they're just strong and resilient, and unfortunately some forms of cancers will just keep coming back. So that's why I think some of our targeted therapies are trying to target the vulnerabilities of the drug-resistant cells, and look even further just beyond trying to target the cancer cells themselves, but trying to target those that are the aggressive subgroups and those that are the drug-resistant subgroups. I know in the leukemia I study, that's a big issue, is drug resistance. The disease can just keep coming back. Patients will go into remission for years and then the disease will come back. So I know I answered your question a little bit, but yeah, no matter how targeted a therapy might be, there's always the possibility that a cancer cell could find a loophole around that, unfortunately.
I'll just mention that your immune system fights cancer. It's gone through cycles of being considered science, to being considered sort of folklore, to being considered science. Anyway, it's currently very, very clear scientifically, and part of the pile of evidence that supports the fact that your immune system protects you from cancer is exactly that, is that cancer cells, now that we've mapped, we've sequenced thousands of genomes of cancer cells, a lot of the changes, abnormalities in cancer cells are to evade the immune system. And there's really no other known function for making mutations in these sets of molecules that people are seeing mutated in cancer cells. They are to evade destruction by the immune system. So I think it's very clear that the immune system is one of the mechanisms your body has to help prevent cancer.
This may sound odd, but we've come into a society where we're so focused on antibacterial this, antibacterial that. Most of us here are of age, where we ate our fair share of dirt and played in the sandbox and ate our sandwich without washing our hands, and we seem to have survived. Are we teaching our kids to be so paranoid of a little bit of dirt that maybe that it's not such a bad thing, a little bit of dirt?
My completely non-scientific opinion as a parent is yes. But, we're here to talk about science and evidence, and that's a hard thing to prove.
I agree with Dr. Marshall. I grew up on a farm, and my kids play in the dirt lots, and they like it. It's good for them.
But there has been real concerted efforts to prove that early education of the immune system, and there's some evidence there. But it's just scientists, we set high standards for ourselves to say something is so, especially if it seems common sense. Although it was common sense at one time that if clearly you want to sanitize your house, that's the best for your baby. And now in society, our mindset is changing, and suddenly our common sense is changing. So, we are suspicious always of common sense, right? And that's my common sense, that kids should live in an environment that's kind of natural, what we evolved in, rather than the modern, but proving it is another matter.
Can anybody explain why antibiotics, excessive antibiotics, are breaking down the immune system?
The first idea is that some antibiotics, when they are administered, are able to kill some beneficial normal flora in the intestine, and then it creates some troubles in terms of immune responses or other things, because homeostasis is dysregulated in that case. And this is one of accepted mechanisms in the field that happens. And there are some certain diseases which are associated with administration of some certain antibiotics, like pseudomembranous colitis or this kind of disease, because they bring some. First of all, they are supposed to kill those harmful bacteria or pathogens, but they also kill some beneficial bacteria from our normal flora, because they are not acting specifically. So then there is a chance to have some invasions in different parts of organs, and we will have immunopathology in those cases.
Antibiotics, that's usually hot topic. Questions? Anyone? So I'll editorialize on that last question and its relationship to the previous one about hygiene, because I think immunology is swung around partly from realization that antibiotics aren't always good for you, to the appreciation of the normal flora, the things that grow inside us, that are normal bacteria. So that somehow the immune system coexists with the fact that we in our colons have large numbers of bacteria, and those bacteria are good for us as we're slowly learning. And that's a very huge and fascinating area right now. Again, so complex that the field is trying to wrap themselves around what are the good bugs that should be in your gut, and what are the bad ones, and do eating probiotics really make a difference, and all that kind of stuff. It's coming. questions? Last chance? Going once? Oh.
Am I right? If you take vitamin D, do you also have to have a good source of calcium and eat that too?
I think that, calcium is definitely important, and vitamin D and calcium sort of, and the parathyroid hormone actually, they sort of regulate each other. So, yeah, taking calcium with taking vitamin D is definitely good as well. So, both are definitely needed.
If you take calcium, do you not have to take magnesium as well?
Chris is the doctor.
So we often offer the immunology topic if you want quickly.
Very quickly. I'll just say that some of these things are actually harder to avoid than you would think because calcium is so prevalent in almost all the foods we eat. But strictly speaking, if you ate 0 calcium, which is technically impossible, but if you did, your vitamin D could still raise your body calcium because it acts on the bones and on the intestine. So 99% of the calcium in your body is in your bones. So if you upregulate your vitamin D and it's not in your gut to bring in, it will get it from your bones. And that's a problem for people who have no calcium intake because their bones get weak eventually. But, so that's, maybe I'll stop there. It's not really, that's as far as the immunology.
Okay. All right, I think I'm looking out in the audience and feeling it myself, a little bit of, brain is full, evening's getting later, this has been fun, but maybe it's time to wrap it up. So I just want to ask you guys to give a hand once more to our